Hello guys and welcome back to Excelsior Academy. So till now we have talked about the mitochondria, chloroplasts, right? Now about the peroxisomes. So they are small single membrane. Now please make sure that you get the difference that it is single membrane. So till now what all are double membrane? Nucleus, mitochondria and chloroplast, right? Where we have seen that it has got both outer as well as inner membrane, and each outer and inner is a bilipid layer. Okay. Now in the case of the peroxisome, it's a single membrane organelle which contains enzymes involved in metabolic reactions. Okay. If you know, your fatty acid degradation also happens in the case of the peroxisomes. So peroxisomes do not have their own genome and all the proteins are coming from the nuclear genome. So यहाँ पे क्या होगा? Then it is going to have its own specific signal peptide so that it gets targeted to the peroxisome. Now, since it is synthesized from the nuclear genome, obviously the proteins are synthesized on the free ribosomes. And then these polypeptide chain, newly synthesized polypeptide chains are then targeted to the peroxisomes. So there are, sorry, however, some membrane proteins are transferred to peroxisomes from the ER. And also like mitochondria and chloroplast, peroxisomes can replicate by division. So this I'll take at the end, like how peroxisomes are formed. So there are two ways by which peroxisomes are synthesized. Okay, so there are two pathways are there. One is by division, the other one is known as de novo pathway. Okay, so we'll come to that at the end, like how the peroxisomes are divided. So here it is, the electron micrograph of a peroxisome. Now, how do you identify in electron micrograph that these are the peroxisomes? Any idea? Like what that special feature they look to identify peroxisomes in the electron micrograph? See, they don't have any color, right? These organelles, except chloroplast, they don't have any colors. So when you observe under the electron micrograph, based on the density, okay, they get this denser region and that lighter region is present, right? Now, based on that outer structure they get, okay, what is the peculiarity about the peroxisomes which they look for to identify in the electron micrograph? So you know that in the case of mitochondria, if you get lines like this, they understand that this is mitochondria. The lines are coming because of the inner membrane, which forms the cristae, right? So that's how they identify this is mitochondria. If it is ER, so they'll see that there's a circular region and जो भी उसके आसपास होगा, dotted. So they say that that is rough ER. Okay. So that's how they understand. But what about the peroxisome? They're just normal circular one. Now tell me, looking at this graph itself, you should be able to get the answer. There is a clear-cut distinction they have given. Krishita, can you identify what is that difference or
lipid vacuole sort of?
no? No, look at this figure and clearly, what is the difference between all these three?
Ma'am, two of them have this dense structure present in between.
Okay. And the third one is not having. That's what you want to say?
Mm-hmm.
Absolutely correct. See, if you see these three structures in the peroxisome at the center, okay, they always find a dense crystalline structure. Okay. Now, this dense crystalline structure is because of the enzymes which they have. So these enzymes, like especially oxidases, okay, as I said, in the case of lysosomes, we have got hydrolases, like, to degrade the stuff and all. Whereas in the case of the peroxisomes, oxidases are there. Examples like peroxisome में क्या होगा, उसका दूसरा काम क्या होता है? Whenever any metabolic reaction is taking place inside a cell, inside a tissue, there are chances that depending on the substrate, sometimes peroxide is also generated, and we know that this is harmful, right? So the enzyme like catalase, the other oxidases, they come and they convert this into oxygen and water. Okay, so these crystalline structure, okay, they are nothing but the dense structure of your enzymes, especially oxidases present. So based on this, whenever a scientist or a researcher sees that there is a circular organelle, something dense is present at the central region, then they identify that as the peroxisome. Okay. Now coming to the functions of the peroxisomes. So human peroxisomes, about 50 different enzymes they have, and they all play important role in carrying out the metabolic activities. Now, peroxisomes were originally defined as organelles which carry out the oxidation reactions which leads to the production of H2O2. Okay, hydrogen peroxide. Now we know that hydrogen peroxide is harmful to the cell. So they also contain enzymes like catalase, which will decompose them into the water and the oxygen. Okay. Now there are a variety of substrates which is broken down by the oxidative reaction. So it's not only the hydrogen peroxide, but also uric acid, certain amino acids, purines, methanol, fatty acids, etc. So इसका क्या काम है? See, if you understand that lysosome is also degrading, then what is the difference? So lysosome का काम क्या है? To get the outside material and degrade that so that the resources are available for the synthesis. Okay, like to provide new amino acids, new DNTPs, RNTPs, etc. अब peroxisome का क्या काम है? Just because of the internal of metabolic pathways which is taking place, जैसे glycolysis हो गया, DNA synthesis हो गया, right? जहाँ पे आपका adenine, guanine, ये सारे synthesis होते हैं. Okay, so when these metabolic pathways are taking place, both anabolic as well as catabolic pathways, there are certain times that these harmful peroxides are generated or uric acid is generated. Okay, so all these are then degraded with the help of the peroxisomes. Okay, so in animal cells, fatty acids are oxidized in both peroxisomes as well as mitochondria. Whereas in the case of yeast and plant, fatty acid oxidation is restricted to the peroxisomes. Now, look at this figure here. So what we are showing here is that there is a fatty acid. Okay. Now, what happens is that when this oxidation is happening, when the fatty acid oxidation is happening, there is a hydrogen peroxide generation. Now, this is harmful if it is not cleaved immediately by the catalase into water and oxygen, then it is gonna oxidize your proteins, यानी कि enzymes. So ये सारे के सारे आपके खराब हो सकते हैं. ठीक है? Did you get this? So whenever there is a fatty acid oxidation taking place in the peroxisome, there is a generation of hydrogen peroxide, but immediately that is being removed with the help of the catalase. Okay? Yeah. Now, in addition to this, okay, providing a compartment for oxidation reaction, they are also involved in biosynthesis of lipids. So here, see, हमने भी पहले क्या देखा था कि आपके स्मूथ एंडोप्लाज्मिक रेटिकुलम में भी आपका लिपिड सिंथेसिस होता है, right? We have seen that in the Golgi also. Now, we are saying it is also happening in the peroxisomes, but it is meant for the specialized type of lipid synthesis. Okay, so they are meant for the synthesis of plasmalogens, which is a family of phospholipids in which the hydrocarbon chain is joined to glycerol by ether bond rather than a ester bond. Now, this plasmalogens, it's really very, very important. Of course, it is a part of the heart and brain, and it is absent in others. So, especially in the heart and brain, it is present. So, it gives what you say strength to the cell. Okay, it provides strength, plasmalogens. Now, please remember this one, because see, we do get confused कि the question आता है. So there was this last time question, not last year, but I don't know which year, but there was this question where they have asked like phospholipids, cardiolipin, plasmalogens, एक और शब्द था sphingomyelin. All these are present, okay? And then the question was asked कि what is synthesized, which among these is synthesized in the mitochondria? ऐसा क्वेश्चन था, ठीक है. सो यू शुड बी एबल टू अंडरस्टैंड कि कहाँ पे कौन सा फॉस्फोलिपिड प्रेजेंट है. सो इन द केस ऑफ द स्मूथ ईआर, इट इज द नॉर्मल लिपिड, ओके, नॉर्मल फॉस्फोलिपिड व्हिच गेट सिंथेसाइज्ड. इन द गोल्जी यू गेट स्पेशलाइज्ड वन जहाँ पे मॉडिफिकेशन होता है, ठीक है. देन उसके बाद आगे आपका माइटोकॉन्ड्रिया में इनर मेम्ब्रेन में ही सिर्फ कार्डियोलिपिन प्रेजेंट है. Now, why people were getting confused? Because cardio बोलने से सबको लगता है it is related to heart. Correct. So that's why this confusion came कि cardiolipin मतलब it is related to heart, and plasmalogens is mainly present in heart and brain. जबकि कार्डियोलिपिन का हार्ट से कुछ लेना देना नहीं है. It is just that it is maximum, it is present in maximum amount in the inner mitochondria. Okay, whereas plasmalogens is present as the major component in the tissues of heart and brain, okay, which is synthesized by the peroxisomes. Guys, did you get the clear-cut difference here?
Yes, ma'am.
Okay. Good. So this is the structure of plasmalogen. This is, I mean, it's okay, no need to remember it. Please just remember that it's just the analogous to the phosphatidylcholine. It's a fatty acid, and instead of ester, you have got the ether bond. Okay. Now this is just the overview, like how in the peroxisomes the fatty acid degradation is happening. Okay. So now right now we won't be going in detail, but just the overviews, because anyways in biochemistry we have to talk about all the steps in detail along with the enzyme structure and function. Okay, so we'll just go a little bit overview so that we understand like how exactly it is happening. Okay, so here you can see that either de novo lipoage. So here also if you see what is happening, the long chain fatty acids or very long chain fatty acid CoA, then यहाँ पे क्या है, it's missing actually, some attached chain of fatty acid is there, and the bile acid synthesis. So all these are happening in the peroxisomes, correct? So what do we see? All these fatty acids, they are actually getting inside of the peroxisomes, right, in the form of the acyl CoA, and it gets involved in the beta oxidation. Okay. Now when beta oxidation is taking place, of course there is a release of heat, acyl CoA, and reactive oxygen species. जब ये reactive oxygen species बनता है, so it means the hydrogen peroxide is part of it. So this is then what to say, normalized, or it is prevented, its harmful effects are prevented by the action of these catalase. So this is what that dense dark image we saw in the electron micrograph. So it's a crystalline core of the oxidases. So they all have these enzymes present over there. Okay, so the ROS is a reactive oxygen species. A part of it is hydrogen peroxide. ऐसा नहीं है कि सिर्फ hydrogen peroxide ही बनता है. There are other radicals also which can form. So all these oxidases and the other, you know, you must be knowing about the SOD. SOD भी सुना होगा आप लोगों ने. SOD, superoxide dismutase, right? So all these enzymes are present here and involved in the ROS metabolism. And of course, then you have got the acyl CoA. So acyl CoA, then it is helping in the other enzymes are there. These enzymes helps in the plasmalogen synthesis. Okay. So here you can see that PMP70 and ABCD1 is given. So for fatty acids and very long fatty acid chains, you have got the transporters known as ABCD1 and PMP70. Okay. Is it clear? The details of the enzymes we'll be studying in the biochemistry portion, so don't worry about it. Yes, any doubts? If you are interested, then you can know or you can study about these enzymes. So they all are, responsible in plasmalogen synthesis. Okay. Like when they have written it as ether lipid synthesis, immediately you should understand that it is meant for the plasmalogen synthesis. Okay. It's your wish to study about these enzymes. वैसे तो इतना detail में जाने की जरूरत नहीं है. But yeah, you can talk about, you can study about it. Sorry. Yeah. Now coming to the peroxisomes in the plants. So first of all, these peroxisomes in the seeds, okay, they're also present in the seeds. And as I said that they're meant for the fatty acid oxidation, and also we know that the seeds are rich in fatty acids, correct? So it is responsible for the conversion of the fatty acids to the carbohydrates, which is providing the energy in the raw materials during the germination. So that's why you know that in the case of the seeds, when we have either it is groundnut lelo, soybean lelo, what is in the maximum cases they are rich in the oils, right? They are having stored fatty acids, and once they start germinating, these fatty acids, okay, they are converted into the carbohydrate. यानी कि क्या? At the end मुझे energy चाहिए, right, to grow, so that I get ATP, and that time. All this conversion is happening in the peroxisomes. Okay. Now, guys, tell me here, these seeds, when they are germinating, are they photosynthetic in nature? Yes. Akshaya, are the seeds photosynthetic in nature while germinating?
I don't think so, ma'am. Right?
Yeah, they are not. They are not. See, first of all, if you see the seeds, that green part is not there. Okay. So it means doesn't have chlorophyll. No chlorophyll, no synthesis. And that time it does not have that, what to say, the accessories which is responsible to carry out the photosynthesis. That time they're germinating, wo grow ho rahe hain. So they are not actually synthesizing. So whatever the stored material is present in the seed, that time everything is being used. Okay. Now, there is one more thing. So people do say that there are many research published which says that we shouldn't be eating, okay, any germinating seeds, because they say that during the germination they do produce toxins to prevent or to escape from their predator. है ना? अब वो अकेले है. Just imagine there is one particular seed is there. It's alone. There is no protection. Okay. So when it starts, पहले तो आपका seed का cover रहता है. Now once that seed cover is being broken because that seed is germinating, it will come out of its coat, right? So now when it is coming out of it, now it becomes little susceptible to all the infections. Are you getting it? And then it starts germinating. जब germinate हो रहा है, कोई चीज पैदा होके इसे धीरे धीरे बड़ा हो रहा है, so that time it's very delicate. So to prevent them from those predators or the infection, they also produce toxins, and that's why people say that we shouldn't be eating germinating stuffs, but we do go for these sprouts. उसको तो हम खूब ऐसे सैलेड में खाते हैं, मटकी बना के, what all is there, right? The chana dal, all these we wait for them to sprout, to become sprout, and then only we have. Sprouting means what? वो भी तो germinate हो रहे हैं ना, हमने उनको पानी दिया, right? So why do we eat sprouts and not the other germinating stuff? If you see in a potato also, if you see that small buds are coming, थोड़ा सा greenish हो जाता है, if you have noticed. Even that, this kind of potato we are not supposed to eat. But हम क्या करते हैं, ये पूरा portion ही remove कर देते हैं, and rest of the potato we eat. Right? They are also germinating part because यही cut करके आप field में डालते हो for the cultivation of potato. So now tell me why in the case of moong dal and chana dal हम उसको sprouts बना के खाते हैं, but in the other cases we throw it. Any idea? Okay, think about it. It's a good question. Okay, and let me know in the evening what will be the answer to this. Just think about it. Okay. The second case is where the peroxisomes in the leaves, they function coordinately with the chloroplast to metabolize the side product of the Calvin cycle. Okay, so later we'll study like how, the in which case we know that the carbon dioxide is getting converted to the carbohydrates during photosynthesis, right? But sometimes what happens is that instead of CO2, oxygen gets involved. Okay, so वहां पे आपका photo respiration हो जाता है. So instead of photo respiration, basically when CO2 is more, normal Kelvin cycle hoga. But if suppose Kelvin cycle is not happening and you have got more of oxygen present nearby the cells, plant cells, okay, तो आप oxygen ले लेते हो. So जब आप oxygen लेते, it goes and combines with the ribulose bisphosphate. So उसको enzyme, what it will do, instead of giving 3-phosphoglyceraldehyde, it will give one molecule of 3-phosphoglyceraldehyde and one molecule of phosphoglycolate. See, जब carbon dioxide combine होता है, you get 6 carbon. Okay, 5 carbon RUBP में जब आपका 1 carbon add होता है, तो you get 2 molecules of 3 phosphoglycerate. Okay, 5 carbon ये हुआ. This is 5 carbon, this is 1 carbon. So you get here 2 molecules of 3 PGA. Now when it is oxygen only, there is no CO2. So you get 1 molecule of 3 PGA. Okay, this is 3 carbon. And here it is 1 molecule of phosphoglycolate, which is 2 carbon. Okay, so total 5 carbon हो गया. अब क्या होता है? This one is of no use to the plant. ये किसी काम का नहीं है. But we will not waste it. What we are going to do is we are going to just provide little bit modification so that this phosphoglycolate can be reused. Okay, so phosphoglycolate dephosphorylation will happen, phosphate group will be removed, glycolate, this glycolate will go to the glycine to the peroxisome, where the glycolate will get converted to the glycine in the peroxisome. Then this glycine gets transferred to the mitochondria, glycine gets converted to serine, serine से वापस आपका glycerate बनता है. Now it is glycerate. So in the peroxisome, what is happening? It is like a shuttle between the chloroplast and the mitochondria. So glycolate को आपने with the help of these crystalline enzymes, oxidases जो यहाँ present है, उससे glycolate को आपने glycine में convert किया. Glycine got converted to serine in the mitochondria. Now this is again shuttle back to the peroxisome where it got converted to glycerate. Now this glycerate can be used. So आप इसमें अभी phosphate group add करो and then put it back into the Kelvin cycle. So this is how peroxisome is playing very important role in photosynthesis. ठीक है, ये अभी हमने ओवरव्यू लिया है. लेटर इन बायोकेमिस्ट्री इन डिटेल वी आर गोना स्टडी अबाउट इट. डिड यू गेट इट गाइस? Yes. Now coming to the peroxisome assembly. So the assembly of peroxisome is mediated by the intersection of the proteins which is synthesized on the free cytosolic ribosomes, and then they are transported to the peroxisomes from the ER. Okay, see here. So either we can have like this. So there are certain proteins. So the proteins known as the PEX proteins or the peroxins. This is specific for your peroxisome. Okay. So these proteins are there. You can see that either in the case of the peroxisome assembly, या फिर ये छोटे छोटे से vesicles बनके all these PEX proteins are present. They can come together and fuse to form a peroxisome. ठीक है. The other proteins can come from the cytosolic ribosomes. So this is one way by which the peroxisomes are formed. Clear? Is this figure is clear? So if you have got these pegs of peroxin proteins, okay, they come together as a vesicle, they fuse together and then they form the peroxisome. Rest of the proteins can be taken by the normal import from the cytosolic ribosomes. ये एक तरीका हो गया. Now the peroxin and PEX proteins, they are transmembrane proteins. Please remember, okay? They are your transmembrane proteins. So they are specific for the peroxisome assembly. So यहाँ पे जो हम ये दिखा रहे हैं, these are your PEX proteins. So the same thing what I have just said right now. So the vesicles budding from the peroxisomal ER, they don't have COP2 proteins, okay? But their formation is driven by the peroxins, the peroxisomal proteins, which just now I have shown in the figure, this one, okay? So ये आपको guide करते हैं for the formation of the peroxisomes. So rather than being targeted to the Golgi apparatus, vesicles from the peroxisomal ER, they fuse to form peroxisomes, in which the matrix proteins can be imported later. तो जब हम इसको peroxisomal ER बोल रहे हैं, what does it mean? So this is a region of the ER where the peroxin proteins are concentrated. ठीक है, तो वहीं पर ही आपका vesicles bud होगा and they will form the peroxisome. So this particular region, okay, of ER is known as the peroxisomal ER. So formation of a functional peroxisome, it requires fusion of two different types of vesicles having the peroxins, okay? Now, peroxisome matrix proteins are translated on free cytosolic ribosomes and later they get imported into the peroxisomes. Now, to get imported into the peroxisomes, again, there is a simple amino acid sequence, okay? This is the signal sequence of peroxisome. It is known as serine, lysine, leucine, okay? This is now your SKL sequence at the C terminus. अब तक हमने क्या देखा था? सारे signal sequence आपके N terminus में थे, right? Because the moment your polypeptide chain is synthesized, N terminal में जो भी signal peptide रहता था, वो आपका detect होता था. Peroxisome में क्या है? पूरा polypeptide chain synthesize होने के बाद at the C terminus you have got this peroxisome targeting signal PTS1. जिसका सिग्नल है आपका सेरिन, लाइसिन एंड ल्यूसिन. प्लीज रिमेंबर, ओके? नाउ इट इज गेटिंग टारगेटेड टू द peroxisomes. This you need to remember. That's why I highlighted in the bold also. The second one is your PTS2. Okay, then it is made up of different sequences of the 9 amino acids. And now this is at the amino terminus. So we have got two signal sequences, that is PTS1 and PTS2. Okay, now PTS1 is made up of SKL and PTS2 is made up of 9 amino acids. PTS1 is present at your C terminus. PTS2 is present at your N terminus. Okay. Now, so PTS1, PTS2, they are both are recognized by a distinct cytosolic receptors, the peroxins PEX5, 7 respectively. So these PEX5 and 7 are present outside in the cytoplasm. Okay, so they are also soluble. Now what is happening is that the PEX5 is going to recognize the C terminal ka PTX1. Okay, then it binds to your cargo protein. The PEX5 and the cargo, this will go and bind to your peroxins, namely PEX 14, 17 and 13, out of which PEX 5 is going to make interactions with the 14, which will result in the membrane pore formation through which the protein can be taken inside. Okay, and the PEX 5 will be recycled. Did you get how the protein is being transported from the cytoplasm to the peroxisome? This figure. Should I repeat? Krishita, is it clear to you or should I repeat?
Please repeat.
Okay. So I hope this much is clear, like what peroxisomal target signal sequence meant. 1 and 2. So 1 is at C terminal, you have got the SKL sequence. For 2, it is N terminals, 9 amino acid sequences are there. Okay. Now, these two signal sequence will be recognized by the receptors known as PEX5 and 7. Okay, they are present in the cytoplasm. They are soluble receptors. Now what is happening is that when in the cytoplasm, you have a peroxisomal protein, okay, if it has got SKL, let's say that it has got PTS1 signal, this will be recognized by your soluble PEX5 cytosolic receptor. ठीक है, यहाँ पे आप देखो PEX5 आया, this purple color is PEX5 which goes and binds at the C terminal. Okay, now this will be recruited to the PEX14, 17, 13. ये क्या है आपके peroxins. अभी हमने last slide में बात किया ना that from the endoplasmic peroxisomal ER these vesicles will be formed. So these peroxins will be present over there, out of which 14, 17, 13 are responsible for the import of the protein from the cytoplasm to the inside of the peroxisome. Okay, into which now what is happening is that PEX5 will interact with the PEX14. Okay, PEX5 plus cargo क्या है यहाँ पे आपका प्रोटीन प्लस PEX14, ये इंटरेक्शन करेगा, because of which what will happen, these peroxins they will make a pore, यानी कि होल सा बनाएंगे, by which they can enter. So the proteins will be entered inside. So you can see that the protein is now entered and the PEX5 which is supposed to remain in the cytoplasm, it's recycled back. Okay, now it is clear. Yeah, this is really very important. Okay, peroxisomes ka till now they haven't asked any disease related questions from the peroxisome, but this is really very important, this figure. Okay, now coming to the peroxisome growth. So as I said earlier, like starting of the lecture, like there are two ways by which peroxisome can be synthesized or it can be grown. Okay, so first one is the de novo pathway. So what is de novo pathway? De novo pathway says that from the start, from the scratch, it is being made, right? So in the case of the peroxisome, you can see when these peroxin proteins are present, these transmembrane peroxisomins or the PEX proteins are present at the peroxisomal ER, they come as a vesicle, they fuse and they form a peroxisome into which the cytosolic protein can be imported. Okay, अब क्या होगा इसके बाद they keep on growing. So this growing means what? More lipids will be added, more proteins will be added, correct? Then what happens, like a bacteria, it will divide. Okay, and then you will have two copies of the peroxisome. So this is how the number of peroxisomes increases in the cell. Now, when does a cell decide that the number of peroxisomes should be increased? It is based on the requirement. आप कितना ज़्यादा टिश्यू, कितना ज़्यादा सेल आपका मेटाबॉलिक पाथवे कैरी आउट कर रहा है, उतना ज़्यादा आपको पेरोक्सिसोम नंबर चाहिए. So based on that, it will keep on dividing. Okay, is this path clear? Okay, now there are certain diseases which is related to the peroxisome. I really want you guys to make a short note on that. Okay, that is Zellweger syndrome. बहुत common है. They haven't asked till now, but yeah, there is nothing wrong in studying all this thing. So Zellweger syndrome, like why it is happening, ऐसा कौन सा enzyme peroxisome का है जो mutate हो जाता है, please study about it. Okay, बाकी इतना कुछ ज़्यादा खास important नहीं है, but this has been asked once, only once, so that's why I'm saying please make a short note on this. Okay, yeah, now let's start with the MCQs. So your first question is, it's really very simple, guys. Okay, which of the following activities are characteristics of peroxisomes? First one, catalase activity, possess a glyoxylate cycle, degradation of very long chain fatty acids and produce dihydrogen. So what is the answer? Okay, let's go alphabetical order. Akshaya, what is the answer?
Ball.
Hmm?
B, ma'am.
Bombay B or Delhi D?
Bombay.
Okay, Akshay is saying 1 and 3. So that is catalyst activity and the degradation of very long chain fatty acids. Krishita and Tanvi, what do you think? The answer is correct?
Yeah, ma'am.
Yes, ma'am.
Absolutely correct. Akshay, your 3 marks. Okay, good. Now, next question. It's for you, Krishita. Which of the following organelle is involved in lipid metabolism?
The peroxisome is involved.
Correct,
Yes.
correct. Because it said lipid metabolism. If it was only synthesis, tab aap,
Synthesis, then SER.
yes, yes.
SER.
Very good. So 3 marks for you. Good. Tanvi, this is for you. Which of the following organelle helps in the penetration of sperm into the egg during fertilization? Tanvi, you there?
Yeah, ma'am.
Would you like to try? So don't think that अभी हमने peroxisome पढ़ा था सारे आंसर peroxisome ही होंगे, ठीक है? What is the answer? Akshay, would you like to try?
I guess it is peroxisome enzyme, the acrosomal enzymes.
Ma'am, I feel it's lysosome.
Yeah, the answer is lysosome. Okay, because see, at the head of your sperm, that acrosomal we talk about, right? So over there, these lysosomal enzymes are present, which is helping in that, penetration, which removes the outer layer of the egg so that it can get the access and it can fuse with the nucleus. Okay, the answer is lysosome. Inside the cell, H2O2 clearance is brought about by dash. Tanvi?
Ma'am, C.
Correct. Peroxisome with the enzyme catalase. Correct. Dash forms the crystalline core inside the peroxisome. Akshaya.
C.
Yes. Highly condensed oxidative enzymes. Last question. Which of the following embedded enzyme in the peroxisomal membrane is involved in the activation of fatty acids before the beta oxidation? Though I have not taken this, but I guess you can guess the answer since I have shown you the figure where in the peroxisome how the fatty acids are entering. So in the fatty acids, what do you think which enzyme is responsible? Activation of fatty acid. Yes, yes. Acyl CoA?
Oxidase?
No.
Synthase.
It is synthase. It is synthase because oxidase means it is going to degrade it, right? But here we need to start the beta oxidation. Of course, we want to digest the fat, but we need to make it activated. Activation के लिए हमें acyl CoA add करना होता है, तो वो काम आपका कौन करेगा? Synthase करता है, ठीक है? Okay, so that's all, guys. So I think this was interesting. So who is the winner? Krishita is the winner. Tanvi, you have to try, okay? It's okay even if you give wrong answer, but you have to try. Okay. So keep it up, guys. ये तो फिर भी बहुत simple questions थे, ठीक है? I'll try to get more questions. Actually, typing थोड़ा सा difficult जाता है. So maybe I can take the photo and मैं वो question ही चिपका दूँगी. That is better for me. So we can do in that way. Okay then, guys. अगर कुछ doubt होगा, let me know in the WhatsApp, and we'll have another session in the evening. Okay, chalo. Have a nice day. Bye bye.
Thank you, ma'am.